This is a production of Cornell University.
Thank you so much for traveling up here for the intros, Carlin, and thank you all for coming. I'm amazed by how many people are in this room. A little bit overwhelmed, especially to see many people with whom I've worked over the years. It's really touching to me that you've all come for this little presentation. So yeah, it's really hard to summarize 42+ years of work in about 42.5 minutes and leave time for a few questions. So I've had to pick and choose, had to make some decisions along the way. What am I going to present? What am I not going to present? What might interest the group? What might tell a little bit of a story that's cohesive? And so my apologies if I've left out some of the work that some of you have been involved in, and I've amplified some other parts of the work that have taken place. There's just no way to fully summarize 42 years in 42 minutes. So, so let's go on and talk about my favorite crop. That's a good, safe place to start. Grapes, they're fantastic. They're amazing. Vinifera grapes, you might be used to seeing leaf types like this on Cabernet Sauvignon, perhaps. But it's such a diverse crop. It's incredibly diverse. There's Vitis mustangensis on the right. Would you recognize that as a grape leaf shape? Tremendous diversity in this genus. Would you recognize Vitis hancockii from Asia as a grape species? Probably not, but this keys out to be a legitimate 38 chromosome grape species. The diversity that you see is just phenomenal. What about Vitis davidii? Also from China, this grape has thorn-like structures. Leaves are more recognizably like grapes, but this has been used in breeding as well. They're not true thorns, but they're thorn-like structures. So not things that you would normally look at and say, wow, this is a grape for sure. Not everything looks like a grape. And the same diversity that you see in leaf shape, in growth habit, in fruit size, color, shape, texture, it just belies the tremendous diversity that you actually can't see. The diversity in the physiology, the diversity in the secondary metabolites, disease resistance that takes some measurements to see. You can't see it all, but just what you can obviously see with your eyes, it just belies the tremendous diversity with this species that we work with that originated between the Black and the Caspian Seas and through the Mediterranean basin, and has, well, the vinifera and Vinifera sylvestris species are from Europe and near Asia, 30+ species in Asia, 30+ species in North America, most of them 38 chromosomes, so tremendous diversity. The goals in our program have focused a lot on disease tolerance, disease resistance. We'll come back to that. Wine quality has always been a cooperative venture with my colleagues in food science. Low temperature tolerance, tolerance early in the season, tolerance in midwinter, tolerance from spring frost are objectives in the program. Of course, you can get some of this and get lousy grapes, so quality has to be an important factor that you breed for. Quality, productivity as well have to be important. So, and you want some ease of culture and management. These are important. We have great resources to work with. This amazingly diverse germplasm. So germplasm collections, wow. I always refer to them as my candy store because I like candy sometimes. So germplasm collections, knowledge of genetics, the molecular marker linkage maps with gene locations that we have nowadays, the sequences of multiple genomes are available to us. And last but really not least, the fact that I am privileged to work with some incredible, amazing colleagues in all fields of science. I think that's been an outstanding aspect of my career, is to have people all around me who just amaze me all the time. So things started back in 1980, and don't worry, I'm not going to spend much time in 1980, but there we are. John Watson, who was a grape breeder and a plum breeder, and the sort of one of the first technicians to work with me. Lucy Morton, a viticulturist who studied in France. Nelson Shawless. Lucy Morton's based in Virginia still. Nelson Shawless, unfortunately, deceased at this point, but a world famous viticulturist. And here I am, 1980. Picture is taken by Pierre Gallet, a famous ampelographer, a famous viticulturist. Unfortunately, he's not in the picture because he took the picture. So these were much simpler times. We go out with notebooks, paper, no cell phones, no tablets. We made crosses between grapes. We chose the best possible parents that we could find. And grapes are like this. No two seedlings are identical. Both parents are highly heterozygous. And the goal in grape breeding was to find the seedlings that combine the best aspects of each parent and leave behind the worst aspects of each parent. So over time, I think we've tried to become much more scientific and deliberate about this process, but I love to share the amazing graphics from my friend and former postdoc, Michael Stream. Give him a little credit for his creativity in creating graphics like this. So one aspect of my program that has been a thread throughout my time here at Geneva has been to breed for disease resistance. So we look for sustainable long-term resistance. And we know that growers spend a lot of time trying to control diseases, a lot of money trying to control diseases. And through the 1980s, we saw loss of fungicides because the pathogens would overcome those fungicides and their effectiveness. But we also knew that grape species had lots of resistance to disease. We knew the genetic resources were there. So how do we utilize them? Downy and powdery mildew are top of the scale in New York. Other diseases are coming in these days with a little bit of climate change, but we focus a lot of time on downy and powdery mildew resistance. And the first thing we did was look at germplasm. At the time, there was no germplasm repository in Geneva until about mid-1980s, I think. It's when the collection was established. So I went looking at germplasm left to me by my predecessors. And it included lots of material that came in from a program that closed down in Illinois, at the University of Illinois, breeding selections and collections from Herb Barrett. And among the germplasm accessions I surveyed was this particular one, Illinois 547-1, a hybrid of Rupestris and Vitis cinerea. And this turned out to be a good choice for breeding work. It was growing under neglect. It was growing in a vineyard that received almost no sprays at all for disease control. And at the end of the season, you go up and find this vine and a few others nearby from other backgrounds, and they're clean, they're disease-free. They're also, you never see trunks like that in New York State, but this one had this tremendous trunk and was growing beautifully under neglect. So that one and a few others were founder parents in disease resistance breeding. And we didn't know much about genetics of disease resistance, but we could sure select phenotypically in the field. And we went to a nursery selection procedure where we would plant out our seedlings from crosses with Illinois 547-1 and its descendants or other grapes that we selected, grow them under no-spray conditions, cut the ones off that had disease, keep the best ones, and plant 10 or 20% to the field for further evaluation in the field. A little close-up of what you see in the nursery. You can see in the lower left, powdery mildew, upper left, downy mildew. And on the right side, here's a vine that was defoliated in the foreground due to downy mildew. The adjacent vine is untouched and of course, marked with a flag. That's the one we saved. And the defoliated types are the ones that were discarded. So very much a phenotypic approach, but still stymied by the fact that we didn't have genes, knowledge of molecular markers, knowledge of chromosomes and where to find traits of interest. So we embarked on a road to develop maps in grapevines. And initially there was collaboration with Norm Whedon. Mary Howell Martins is here in the back. So Mary Howell was a part of this early work on isozyme genetics in grapes. Well, eight isozyme loci, 3 of them, there was 3 pairs, first linkages. Well, I say likely first linkages because as soon as you say it's the first linkage, someone will say, oh no, there's another publication somewhere that also reported linkage. So likely the first linkage groups reported in grapes. And then by 1995, worked with Mohammed Lodi, a student of mine, also teaming up with Norm Whedon, a first molecular marker-based linkage map with mostly rapid markers, RAPD markers, 16 RFLP and isozyme markers as well. This was the beginning. So likely a first molecular marker map in grapes. There's Norm in the middle between Susan and I. He was here a couple of weeks ago for a little visit. So Norm left for Montana State University a number of years ago, but we still stay in touch and glad that he was able to stop by. By the late 90s, we had identified a locus coming from that Illinois 547-1, REN2, sourced from Vitis cinerea, a locus for powdery mildew resistance. And so 1998, locus identified, 2001, we had a marker-assisted selection procedure. Yay, 1 locus, 1 gene, 1 little bit of information, and we hungered for more. So a turning point in our program was the access we were given to RUN1 and RPV1. RUN1 for powdery mildew resistance, RPV1 for downy mildew resistance. These are genes that came from the muscadine species, a 40 chromosome species from southeastern United States. It was backcrossed by colleagues in France, like Alain Bouquet, into a vinifera background, into a 38 chromosome bunch grape background, and later further backcrosses into wine grape backgrounds adapted to Hungary were made by Paul Kozma. And Paul was kind enough to share seed with me, from which we selected individuals that had the RUN1 locus. And RPV1, same locus, same location in the genome. So this was really pivotal to us because these are 2 of the best genes that we work with for downy and powdery mildew resistance. And when you look at these plants that descended then from the original seedlings of Agar 9911 from Hungary, you can see in the upper left an unselected population, in fact, it's a population without the RUN1 gene, devastated at the end of the season by disease. And the other 3 panels are all vines carrying the RUN1 locus for powdery and downy mildew resistance. And you can see these vines under no spray conditions are thriving pretty nicely. So later in the process, we did adopt marker-assisted selection as there were more and more genes to work with. A gene like RUN1, we can pick this out phenotypically, but we had no idea which genes had 2, 3, or 4 different loci for resistance. And the idea was to try to stack the genes, to try to put off the possibility that a pathogen would mutate and overcome a single gene by having multiple genes in the same vines. So through marker-assisted selection, we would plant out nursery vines, and now they would look quite nice, quite good. So the predictions being made by the markers were quite accurate, and these vines were really doing well in the nursery. So this is all along the way of breeding hybrid grapes in the program. Yet there are a lot of vinifera grape growers of Chardonnay, Pinot Noir, Riesling, et cetera, Thompson seedless around the world. And what could we do for those growing vinifera? Our idea was to use Transgenics to use the biolistic process that had been developed here at Geneva by my colleague John Sanford, Ted Klein, and many other cooperators, and do some gene transformation experiments to check whether we can get some genes for disease resistance into vinifera grapes, with the idea being to try to reduce sprays. Hybrid grapes typically take 4 to 7 sprays per season. Vinifera grapes 10 to 15 sprays per season. So could we do something for vinifera growers without changing the characteristics of the original vines? And Julie was with me through many of these years, Julie Kickert, Pat Wallace, and Jose Vidal, and a host of others. We had I believe this was a real first, the first field trials of transgenic Chardonnay and Merlot here in Geneva, and also at a location in California. And the upshot of all the work to insert endochitinase from trichoderma or antimicrobial peptides into vinifera grapes was that we had some small elevations in powdery mildew resistance, no resistance to Guignardia, which was black rot. Under certain circumstances, we tried this bunch rot resistance and crown gall resistance, some reduction in crown gall resistance. So we were able to measure resistance to bacterial and fungal pathogens. But none of this seemed to be at a level that would justify commercialization and deregulation. The resistance levels weren't great enough, and the demand from the industry was on and off, on and off. The funding was both on and off. There were many people who were hesitant to adopt transgenics. So we eventually ended this project in the mid-2000s, 2008 or 9 or so. We no longer continued, but we did continue a lot of the work in molecular mapping. So molecular mapping, gene identification, techniques that we could use to apply to hybrid grape breeding. So what could we do to really improve hybrid grape breeding? And now I'd like to focus on the last 11 years or so, since the VitisGen 1 and VitisGen 2 projects came our way. I'd like to tell you a little bit about the work that's been done a little more recently. And this has been a collaborative project with Lance, co-lead on these projects, Lance Cadel-Davidson, and 25 investigators across the country in each of these projects. Tim Martinson, Anna Catherine Mansfield. We have a number of investigators who have been on these projects in Geneva, Ithaca, Davis, California, Minnesota, Missouri, across the country. So let's talk a little bit about these 2 SCRI projects. They have been taking place since 2011. The second one ended in August this year. The total funding for the two projects was about $11 million over that period of time, funded by the Specialty Crops Crop Research Initiative. And I'd have to say I had my doubts going into the project, whether you can get teams of 25 people across the country working in sync together. And we did. That was one of the most gratifying things, is to see the progress we could all make together, given a good level of funding and some, again, amazing colleagues who were putting their minds to the tasks at hand. So we sought to apply technological innovations in phenotyping and genotyping to deliver breeding lines with powdery mildew resistance, especially durable resistance, along with high fruit quality. And along the way, we investigated a number of other traits as well. So one of the key innovations in the VitisGen 2 project was a new marker technology, ramp-seq technology, which was commercialized with integrated DNA technologies. And Lance and others had the idea of developing haplotype-based SNP markers based on sequencing a 200-300 base pair sequence and such sequences throughout the genome, and finding sequences of 4 or 5 single nucleotide polymorphisms as a signature of a particular marker. So you're looking at a marker sequence as an allele. And as soon as one SNP is different, it's a different allele. So the other thing about the markers developed is that they're based on the core genome. They're based on genes that are in common among a diversity of grape cultivars and species. So North American species, vinifera. So the idea was to develop markers that were well anchored, transferable, and covered the entire genome. So you can see visually the improvement in marker technology going on the left from SSR-based maps or microsatellite-based maps to genotyping by sequencing maps done in VitisGen 1, dense markers not so easily transferable from population to population. And going on to maps that have 99% coverage of the genome, an average distance between markers of under a half a centimorgan. So those are the ones on the right, covering, incorporating both ramp-seq as well as GBS markers. Another key component in VitisGen 2, Dave Gaduri, Lance, colleagues at RPI, Rensselaer Polytech, lighting specialists, had some ideas about how to improve on the VitisGen 1 approach, which involved a lot of microscopy to analyze powdery mildew resistance and looking at high-fold transects under a microscope. And this takes hours, lots of eye fatigue, lots of time to carry out this project. And how can you do this a little more rapidly and phenotype even just as objectively, but much more rapidly? And they came up with this brilliant solution, a robotic imaging system, non-destructive. You could image leaf discs that have been inoculated with powdery mildew on successive dates, 1 day after inoculation, 3, 5, 7. You can and you can analyze the images. You can take pictures of the leaf discs, analyze the images automatically using a convolutional neural network. Now they're way beyond that in collaborations with Yu Zhang, using artificial intelligence to pick out hyphae. But this was a great innovation. Blackbird, the more advanced prototype is shown here in the lower left. And just a little diversion from this train of thought. before I go into some elements of marker-assisted selection. So we did a wonderful project to better understand the flower sex locus in grapes. And we determined that there are actually two different hermaphrodite alleles. Let's see if I can use the pointer up here. So in this project, we know that female flowers of grapes are basically dioecious. Wild vines are either male or female. Females are basically homozygous for an F allele. Males are heterozygous, MF. And in North American, East Asian, and European accessions, you can analyze over 1,000 SNP markers across the sex locus and get basically the same genotypes for your males and females. So in this project, they found two different hermaphrodite loci. And the more important result for me as a breeder is the development of really excellent markers for flower sex. So we could not use some of the markers that had been developed previously in the North American type germplasm we've been using, but these markers have been working really well in the marker-assisted selection pipeline for us. So VitisGen 1 and VitisGen 2 provided marker-assisted selection pipelines to grape breeding programs across the United States, including ours. And in the early days, it was pretty simple. A couple of different genes of interest, like RUN1, REN2. Are they present? Are they absent? Which ones have both genes? Okay, save the ones with both alleles, discard the ones that have neither allele, or that have REN2 only, or RUN1 only, discard if they're negative. Very simple decision-making. Now decision-making is a little more complex, but now based on ramp-seq markers, first of all, we can pick out the selves, get rid of self-pollinations that occur. Those are the -1s here. We can pick out markers for male loci. We don't want the males, but locus 254, locus 18, they come from a parent from the O block. Those are designated for the male locus. And when they occur among the seedlings, those are discards. And the opposite is true for RUN1. When locus 13 or 8, those are the ones in blue, or locus 1 for this particular marker. When those occur in the seedlings, those are the ones we want. There's an F statistic probability that tells us which seedlings are the likely ones to carry RUN1, which are not likely to carry RUN1. So that's a sample of the spreadsheet. The spreadsheet goes for a couple of miles this way or that way if you're looking at the screen. Lots of markers, lots of loci that we're working at, loci for powdery mildew, downy mildew resistance. So let me go through some of the loci and other advances that were illuminated through the VitisGen project. And this is really fun to do because, I'm only going through a portion of what the VitisGen project accomplished through a breeder's lens, through my own eyes. So in biotic stress resistance, one of our goals was to facilitate breeding for resistance, especially powdery mildew resistance, across the country. So we developed lines that were studied by Surya, by Lance's group, David's group, to look at to understand the interactions between multiple loci. But also we created what we call RenStack1 and RenStack2 lines to develop single individual clones that had all 4 loci. So for instance, I made a cross with one of my RUN1, sorry, that's RUN1 and REN2, RUN1, REN1 hybrids. I made a cross of that with Vitis piazetzkii pollen supplied from Samara and Andy Walker's lab. and Piaseski carried Ren 6 and Ren 7. So we wound up with several individuals that harbored all 4 loci, were tested by markers to carry all 4 loci. And the same thing happened with a collaboration between Craig Ledbetter at Parlier, California, a table grape raisin grape breeder, and Matt Clark at the University of Minnesota. Another cross that they made carried 6 different loci for powdery mildew resistance. 4 individuals from that cross, 4 individuals from Ren Stack 1 are now with Foundation Plant Services, going through indexing protocols to be available as pollen or as cuttings to breeders across the country, so they can have immediate access to these loci, all in one place, one pollen source. So now, as we improved phenotyping and genotyping, we had great maps. In VitisGen 1, Steve Luce brought some information about segregation of Phomopsis resistance to a group meeting one day, and Paula Barba had good genetic maps for those populations. And by that afternoon, she had located through GWAS, she had located a gene on chromosome 15 from Vitis cinerea and another on chromosome 7 from cultivar horizon for Phomopsis resistance, 2 good qualitative loci for Phomopsis resistance. So that's how we put phenotyping together with molecular mapping. This was a study, now the next one's a study I wasn't a part of myself. This is within the VitisGen 2 project. Lance and others had been working, and Dave Ramming had been working for years to try to understand segregation for powdery mildew resistance, descended from Tamiami, a Vitis estivalis type source. And eventually they found the Ren 11 locus on chromosome 15 for resistance, and very good effective resistance on leaves, rachis, berries, and often on stems as well. In another study also, this one I'm a little bit a part of, a cross with Vitis amurensis grown at the McCarthy farm here. Lance, Soria, and team, probably not mentioning all the people that should get credit, but there was a determination that there was a Ren 12 locus on chromosome 13 from Vitis amurensis, a new locus. Both of these genes have now been incorporated into the Cornell Geneva breeding program. We're excited to be looking at their progeny in the field and coming into fruit in the future. This was a study now, another study in a Vitis amurensis hybrid population. Sorry, yeah, going back to Ren 12, that was actually a cross made in California, grown in California, but this was the amurensis population grown here in Geneva, and work done by Lance, Soria, and others located some new genes for downy mildew resistance. And again, I've also tried to incorporate these loci, particularly the new ones on chromosome 13 and chromosome 10, the ones that show up multiple times in different experiments, to try to incorporate those into adapted material in the breeding program. So markers for fruit quality, a little more difficult to find markers for fruit quality, but through persistence and multiple years of studies, collaboration with Gavin Sachs, Anna Catherine Mansfield, Noam Reshef, who is now back in Israel as a grape breeder. We did determine in this one particular study the genetic basis of anthocyanin acylation in North American grapes, and there's a major locus for acylation of pigments. And acylation is important in color stability in wines. So this marker has now been implemented in breeding programs. Another study that covered a range of characteristics, but I'll focus here on one particular characteristic, and that is the six carbon compounds, hexanols, hexanals. They're responsible for some of the grassy green odors that you find in some hybrid grapes. And in this project on chromosome 2, there was a locus associated with hexanals. There's still more work to do to utilize this locus in breeding programs, but it's a first indication that we might be able to pick out, well, this would be great if we can pick out some fruit quality traits years before we ever see fruit. So we're making progress in those directions. In malate, there was a multi-year study, again, with Gavin, Anna Catherine, Avi Karn, Dave Mans, Lance, Noam Reshef led this project for several years. And we studied malate levels in populations here at Geneva and some other populations in the VitisGen project. And we located loci controlling acidity or malate levels on chromosomes 1, 7, and 17. However, these were not so transferable to unrelated populations. And in fact, if you look through the literature, you'll find malate levels are associated with genetic loci on all 19 linkage groups. So this seems to be more of a very quantitative characteristic with different forms of control in different genetic backgrounds. We've looked at the order of bud break, phenology of grapes, genetic studies of growth stages, genetic studies of bloom timing. We've carried out in the last couple of years cooperatively with Katie Gold, studies using hyperspectral analyses of leaf and fruit traits. And there's too much here to go into much detail other than to tell you that we are interested in some of these climate adaptation related traits and the order of bud break that may help us avoid problems with spring frost. So I wanted to mention that those studies were very recent and not yet summarized, but also I want to spend the last few minutes on the products of our program and make a very important point with you that all of the work we do is only important if we can justify it in the field, if we can ground truth it, grow grapes in the field, and see the results and see whether what we predict based on lab results and marker results is really true in the field. After all, if we are to name and release a new grape, who will grow it if we have no field data? Someone will be making a 30-year investment, but they'll have to take our word for it. They would rather take data. They would rather know that these do what we say they're going to do based on field testing for multiple years, multiple locations. And through the project, we have a lot of really interesting seedlings. We're starting to make wine from some of them. Some of them look very promising for wine quality as well. These, to me, this is the part that's going to be hardest for me to leave behind when and if I retire. Because I think that these VitisGen related seedlings growing here under no spray conditions hold lots of promise for the future. If we were to review the timeline of grape releases from the program, we had releases in 1981, 82. Melody was of some commercial success. Einstein Seedless went to a number of home gardeners. Chardonnay was quite successful in Missouri, Michigan, Arkansas, and really was released at the request of breeders, not breeders, but viticulturists in Arkansas and Michigan. Aramella, Arundel, more recent releases, 2013. Aramella is taking off a little faster than Arundel. Arundel being the first grape that came out of our no spray breeding program, our disease resistance breeding program. New York 81 next year, other thing coming soon. And Traminette, a 1996 release, very impactful. Hundreds of thousands of vines have been sold. Excellent wines, award-winning wines have been made. It's a hybrid with Gewürztraminer. And we see lots of wineries in New York State making really excellent wines with Traminette. By the way, has late bud break, helps avoid some spring frost issues. Now, a little comment about one of our more recent releases. My inspiration comes from some of the work on developing tetraploid grapes, 76 chromosome grapes, not just 38 chromosomes. 76 chromosomes is associated with having increased cell size, increased leaf size, but also increased fruit size. So Japanese grapes are amazing. They're large, they're flavorful. And years ago, we made some crosses with some Japanese grapes, crossed with some of the tetraploids that I had inherited in the breeding program at Geneva. And we developed our own tetraploid, a naturally seedless grape, Concord-like with 5 to 7 gram berries, about twice the size or twice the weight of a Concord berry. So a nice little impact of breeding at the tetraploid level for visually appealing, large berried seedless grapes. Sometimes the numbers surprise us from the more recent releases. You can look at the annual wine value, and it's nice to know the impact that we're having. These are back of the envelope type of calculations, but Valvin Muscat having pretty good impact, Noiret having a pretty significant impact, over close to $19 million worth of wine sold annually based on numbers of vines sold, based on acreage planted. Aramella, Arundel, released in 2013, so you wouldn't expect that to be fully reflected in numbers that go from 2010 to 19, but we're seeing a 50% increase between this period and 2020-22. So I think Aramella is off to a really good start. And you know, I think we've done well in giving the industry some good grapes to work with. And I'm hoping I've done well to leave a program in good shape for the grape breeder, grape geneticist of the future. There is a search that's ongoing in horticulture for a new grapevine breeder, geneticist, genomics person. And I think this person has a lot of opportunities. They will have multiple selections with stacks of genes, resistance genes, introgressed into a cultivated background adapted to New York and Eastern United States. They'll have full ramp-seq marker sets for everything in the program. That's about a 2,000 marker set for everything in the program, plus phenotypic data for a number of biparental mapping populations. They'll have access to an endowed fellowship in grapevine improvement, royalty income account with some tremendous amounts of money in it. They'll have immediately the opportunity to be part of this team, some of whom are in the room, some of whom are in Ithaca, others across the United States. But immediately they become part of this fantastic Cornell USDA team of grape and wine scientists. Accommodations would be available in the soon-to-be-built, we hope, National Grapevine Improvement Center to open its doors, hopefully in about 2026. And the VitisGen 3 project was funded, led by Matt Clark and Lance Cato-Davidson. Matt Clark at the University of Minnesota. I hope they'll have an opportunity to join VitisGen 3. Way too many people to thank, to acknowledge. Had fantastic students, technicians, postdocs, research associates, amazing colleagues, too numerous to name individually, great scientists on the VitisGen projects. And I'm going from these days in the 1980s to these days, spending a little more time with family, some time, but a little less time with grapes. I have all of these people to thank as well, the people in the upper two panels, great supporters and off to great starts in their lives as well. And all of you. And so let me just wrap up there and thank you all.
Anybody from Ithaca have questions?
I've got a question, Bruce. Steve Reiner's here.
Hi, Steve.
I'm sorry I couldn't be there in person. And again, I just wanted to thank you for everything over the years. When I first came to Geneva, you were one of the people in your family. We met with you and you sold us on coming there. Our families continue to be close. Our wives have worked together for years. Our sons work together, continue to do that. So again, it's just been really good. And you and I served as Little League coaches together. So you really get to know somebody when you do that. But I wanted to put on my chair's hat, my former chair's hat for a second, and just thank you for being such a good department citizen. It was obvious when I took over as chair, you did not want to be chair, but you offered your help in any way you could, and you were good on your word, whether it was committee work, whatever I asked of you, you always were willing to do it, and you did a great job with it. And sometimes we don't always appreciate how important it is to be a good citizen in our departments, but you are the best example I've got of that. And I just wanted to thank you for all of that, and good luck in your retirement.
Wow. So Steve, the check is in the mail. Thank you. And you're too kind. And I thought you said you had a question.
I do have a question. So after 40 years, what would you have done? What one thing would you have done differently, do you think?
Oh my God. You make too many mistakes along the way, and you got to admit to making mistakes. Jeez. But when I think about things I regret, God, I really don't want to go into those. Saying the wrong thing to the wrong person, like the check is in the mail. You know? No, if I were to do things a little differently, I think I would have followed my instinct and my training and started disease resistance breeding 5 years earlier. I think I waited a little too long, because I think it takes a long time to breed new grapes. And I think I would have been further along if I'd started 5 years earlier. And the rest of the things I'm thinking of, I'm not going to say.
Bruce, great talk. Certainly, we are all excited about what you have accomplished. I have one question for you. That is, if one of the if the successful candidate stand here today, what one thing you want to say to her? Oh,
Thank you.
And to them. I would say, don't let me tell you what to do. Trust your training. Take advantage of opportunities. Please don't try to go it alone. By the way, I hope I turned on the ceiling mic, so I hope they heard you in Ithaca. I would say collaborate. Collaborate is really important. I think most of us may be here in this room, but maybe not everyone recognizes the importance of being part of the team and collaborating, finding people with complementary skills, and find some good colleagues to work with.
Any questions from Ithaca?
We have a couple more over here.
Yeah, go ahead.
Well, Bruce, you suggested that these grapes are making very nice wines. But where are they?
No,
Couldn't believe you.
did you No, I.
Send us a check.
Oh, wait, Alan was invited to wine tastings back in September. So.
I had COVID then.
Oh.
We didn't want to.
Okay,
I got
well, when there's a
I'll
repeat
stay.
tasting, we're still on the list.
I think there are neglected opportunities for making use of North American Vitis germplasm.
I think we've only begun to tap the resources in Vitis germplasm. I think even if we've covered screening a number of different species, there's such diversity within each species. I think maybe we've uncovered certain alleles from each species, but shouldn't there be multiple alleles at the same locus, perhaps, within a species? So I think the identification of new loci is only limited by the needs of breeders, the identification of funding to screen for those traits, and then the identification of further loci within species. There's a lot of untapped resources. So thank goodness for the germplasm repositories that we have to preserve the resources that are out there. Gennaro, oh, sorry.
Yeah. So for table grapes and perhaps even wine grapes, we've seen these developments of candy cane and which is moon drops and all of these very interesting new flavors that have come out. Have you experienced any of that in your breeding program and participate, for wine and for table grapes?
Yeah. Well, I have the room mics on, but yeah, the question is about whether we've experienced some of the inroads made into the table grape market with grapes like cotton candy or witch's fingers or candy drops, things like that. And also in the breeding program. So yes and yes. Yeah. First of all, a lot of the unusual flavors that are coming through in the new grapes from International Fruit Genetics in California, they're sourced from Vitis labrusca. They are components of labrusca grapes being not inserted, but through crossing, bred into new cultivars with vinifera-like qualities, textures, characteristics, good storage, shipping characteristics, but flavor from labrusca. So cotton candy is a big example of that. When we also have people from industry, from California industry, let's say, taste some of our grapes, or people from Europe, they sometimes identify some new characteristics that they find unique and interesting and useful. Chocolate maybe pops up as one of those words in some of the wine grapes. Some of the floral characteristics are unique. But yeah, we do get that comment from people from different programs that taste our wines. And they're also finding certain unique traits and trait combinations in our selections.
That is very.
Yeah. Do we have time for?
Yeah, just one.
Okay, well, Tim has a question.
Yeah.
So Bruce, I became aware of the something beyond marker assisted selection, and that's genomic selection. And I don't quite understand what that means and how it would be a different approach. Is that the next frontier to be crossed? Or what advantages or disadvantages?
So, yeah, I think genomic selection. So the question is about marker assisted selection, whether the advantages, disadvantages, is that the next frontier? I think that may be part of the story in the future, predicting breeding values, helping pick out parents based on more than the phenotypes and the knowledge of what markers each parent carries. That's been important for me to know phenotypes and markers, what genes for resistance or acylation are carried by different parents. Genomic selection provides a lot of opportunity that even some smaller breeding programs are now taking advantage of sweet potatoes, I think we'll hear about later today or tomorrow. Blueberry breeding we heard about recently. The idea is to use thousands of markers, be able to use those to predict certain quantitative characteristics, and thereby utilize the information to predict which seedlings should be saved for some of the more quantitative traits. We've gone for what we call the low-hanging fruit in much of the last 20 years, the single genes that have a big impact on the program and combinations of those single genes. But I do see that there should be a role in some of the, going into the future with genomic selection and genomic prediction. But the grapevines are still large plants. We can't grow so many thousands. And as you get into projects that are looking at traits that are controlled throughout the genome, I think you need to further increase population sizes and find ways to do that. There are still cost-effective, affordable, to really be able to implement. There are going to be some practical hurdles to implement, but not out of the question.
Thanks.
This has been a production of Cornell University, on the web at cornell.edu.